The period before birth is the foundation for all human development. Pregnancy begins when egg and sperm cells unite and exchange hereditary material. Eventually, the sperm and egg are transformed into a living, breathing human being. We will look at the three main periods of prenatal development, some problems that can occur during development before birth, then focus on birth and the new family. Let's begin with a brief review of genetics. First, the universal. All living things are composed of cells that promote growth and sustain life according to instructions in their molecules of DNA. DNA is a molecule that contains the chemical instructions for cells to manufacture various proteins and promotes growth and sustains life. When a cell divides, one of its main jobs is to make sure that each of the two new cells gets a full perfect copy of genetic material. Mistakes during copying or unequal division of the genetic material between cells can lead to cells that are unhealthy or dysfunctional. When a cell in the body divides, it will pass on a copy of its DNA to each of its daughter cells. Physically speaking, DNA is a long string of paired chemical units that come in four different types, abbreviated A, T, C, and G, and it carries information organized into units called genes. Chromosomes are molecules of DNA. If you took the DNA from all the cells in your body and lined it up end to end, it would form a strand 6,000 million miles long. To store this important information, DNA molecules are tightly packed to make structures called chromosomes. There are 46 chromosomes arranged in 23 pairs. A gene is a length of DNA that codes for a specific protein. Now, genes typically provide instructions for making proteins, which give cells and organisms their functional characteristics. Genes influence how we look on the outside and how we work on the inside. They contain the information our bodies need to make chemicals called proteins. The genes on the chromosomes in the nucleus of each cell instruct the cell to manufacture particular proteins. The code for a protein is a combination of four bases, TAGC, thymine, adenine, guanine, and cytosine. Proteins are large, complex molecules that play many critical roles in the body. They form the structure of our bodies, as well as playing an important role in the processes that keep us alive. They do most of the work in the cells and are required for the structure, function, and regulation of the body's tissues and organs. Life begins with gametes that combine to produce a zygote. Gametes are reproductive cells, sperm and ova. Each gamete consists of 23 chromosomes. A zygote is two gametes that combine, sperm and ovum, to produce a new individual with 23 chromosomes from each parent. Even though both are important in the process of fertilization, there's a big difference between a gamete and a zygote. A gamete is a reproductive cell that plays a critical role in the process of fertilization. Produced by organisms that sexually reproduce, a male gamete, also called a sperm cell, fuses with a female gamete, also called an ovum or egg cell, to develop a zygote. This ovum is about to become a zygote. It has been penetrated by a single sperm whose nucleus will move to next to the nucleus of the ovum. Soon, the two nuclei will fuse, bringing together about 20,000 genes to guide development. It's important to remember that overall, many genes are identical for every human being. Some genes, though, vary slightly in their codes from one person to another. An allele is a variation of a gene or any of the possible forms in which a gene for a particular trait can occur. The effects of variations vary greatly from causing life-threatening conditions to having no detectable effect at all. An individual inherits two alleles for each gene, one from each parent. Now, this is a bit of an oversimplification, as there can be several genes involved in the expression of a particular trait, but this is a good way to understand the basic concept of dominant and recessive alleles. For example, the allele for brown eyes is dominant. Therefore, you only need one copy of the brown eye allele to have brown eyes. Although with two copies, you will still have brown eyes. The allele for blue eyes is recessive. Therefore, to have blue eyes, you need to have two copies of the blue eye allele. And you can see from the graph on the left, we have a father who has brown eyes and a mother who has brown eyes, and both are heterozygous for that particular trait. So it takes a particular combination of alleles for the offspring to have blue eyes. On the right, the father has blue eyes, and he is homozygous for that recessive allele. The mother has brown eyes, and she is heterozygous for that trait. And you can see the combinations that need to occur for the offspring to have blue eyes. We know that genes are passed down from generation to generation. Genotype and phenotype are very similar sounding words that are related but actually mean different things. Genotype is an organism's genetic inheritance or genetic potential and is unique for each organism. Genotypes can only be determined by biological tests, not by observations. Phenotype describes the observable characteristics of an organism, including appearance, personality, intelligence, and all other traits. Phenotype is what you see, the visible or observable expression of the results of genes combined with the environmental influence on an organism's appearance or behavior. The huge variety of people's faces reflects each person's genetic individuality. Genetic diversity is important because it helps maintain the health of a population by including alleles that may be valuable in resisting diseases and other environmental stresses. Maintaining diversity gives the population the flexibility to adapt. If the environment changes, a population that has a higher variability of alleles will be better able to evolve, to adapt to the new environment. Now, even though there are individual differences, there still is a lot of similarity. The genome is the full set of genes that are the instructions to make an individual member of a certain species. The similarity between two people is 99.5%. Similarity between humans and chimpanzees, 98%. Similarity between humans and every other mammal, 90%. The Human Genome Project was the international collaborative research program whose goal was the complete mapping and understanding of all the genes of human beings. It found about 20,000 to 23,000 genes in humans. The exact number is not known. Each egg and sperm has 23 chromosomes. When a sperm penetrates an egg, their chromosomes combine to produce 23 pairs of chromosomes. The first 22 pairs of chromosomes are called autosomes. The 23rd pair determines the sex of the child. When the 23rd pair consists of two X chromosomes, the result is a girl. An X and Y chromosome produce a boy. So humans usually possess 46 chromosomes, 44 autosomes, and 2 sex chromosomes. This arrangement means that it is the male that determines the sex of the offspring when fertilization occurs. Genes on the X chromosome are said to be X-linked. X-linked genes have distinctive inheritance patterns because they are present in different numbers in females, XX, and males, XY. X-linked human genetic disorders are much more common in males than in females due to the X-linked inheritance pattern. If a male inherits an X-linked recessive trait from his mother, he expresses that trait because the Y from his father has no counteracting gene. Females are more likely to be carriers of X-linked traits, but are less likely to express them. The textbook uses the example of color blindness as an X-linked gene that can be passed on to offspring. An X-linked recessive gene almost never has a counterpart on the Y chromosome. Therefore, recessive traits on the X chromosome affect sons more than daughters. Genetic interactions are complex processes. We know that almost every trait is polygenic or affected by many genes, and multifactorial, influenced by many factors. Each cell expresses or turns on only a fraction of its genes. The rest of the genes are repressed or turned off. The process of turning genes on and off is known as gene regulation. Gene regulation is an important part of normal development. Genes are turned on and off in different patterns during development to make a brain cell look and act different from a liver or muscle cell, for example. Genes can't control an organism on their own. Rather, they must interact with and respond to the organism's environment. Although we know that the regulation of genes is critical for life, as genes create proteins, this complex regulation process is not yet fully understood. Proteins form the intermediate point between our genes and our behavior. Researchers have found new evidence of how certain proteins are working at the molecular level, paving the way for new improved drugs to treat psychiatric disorders. Are all people either short or tall, fat or thin, very dark or very light? It seems like a silly question, but this is the way that many people think of genes and traits. Very few phenotypes or observable traits are all or nothing. For example, people don't just come in 2 or 3 colors, as we would if skin color was as simple as a single dominant or recessive gene. Instead, other factors come into play in determining our characteristics. One factor is additive genes, which are alleles that contribute to traits such as height, weight, color, hair color, eye color, and complexion. Since humans usually inherit one copy of each chromosome from their mother or father, most people have 2 alleles for each gene. But there can be more additive alleles for a given gene. In fact, the number of copies of an additive allele determines if you will have an observable trait. It's important to note that polygenic traits and phenotypes cannot be distinctly categorized. Rather, they exist on a spectrum. There's no black or white, but instead black, white, and countless shades of gray in between. That explains why there's a wide array of hair color, skin color, and eye color, as well as countless shades of those colors. A carrier is a person whose genotype includes a gene that is not expressed in the phenotype. The unexpressed gene occurs in half of the carrier's gametes and is passed on to half of the carrier's offspring. The offspring can be a carrier or express the gene in the phenotype. While approximately 99% of our DNA is the same among everyone, that 1% can mean a lot of variation in a person. We know that every person carries 2 copies of most genes, one from each parent. A carrier, also known as a hereditary carrier or a genetic carrier, is an individual who has a mutation in one copy of a gene. While as a carrier, the person doesn't have the disease associated with the mutation, he or she may pass this mutation on to his or her children. Current research has shown a genetic mutation to be linked with schizophrenia, bipolar disorder, and major depression. Being able to identify carrier genes and understanding how they work is important in developmental psychology. Researchers led by the University of Edinburgh scanned the brains of people that carry a specific genetic mutation. The researchers showed that carriers of this mutation had lower levels of a chemical called glutamate in certain areas of their brain. The mutation can cause part of one chromosome to swap places with another. The expressed mutation then results in a genetic interaction and disruption of a gene that is associated with schizophrenia, bipolar disorder, and recurrent major depressive disorder. Experts say these findings could help in the quest for new treatments for these particular psychiatric disorders. Let's move on to the next section of the chapter, From Zygote to Newborn. The many changes that transform an egg fertilized by a sperm cell into a newborn human constitute prenatal development. Prenatal development takes an average of 38 weeks, which are divided into 3 periods: the germinal period, the embryonic period, and the fetal period. The shortest prenatal period is the germinal period, the first 14 days. The zygote begins duplication and division within hours of conception. The teaspoon or so of seminal fluid produced during a fertile male's ejaculation contains from 200 to 500 million sperm. Of the sperm released into the vagina, only a few hundred complete the 6 or 7 inch journey to the fallopian tubes. An egg arrives there monthly, hours after it is released by an ovary. When one sperm finally penetrates the cellular wall of the egg, chemical changes occur in the wall immediately, blocking out other sperm. Then the nuclei of the egg and sperm fuse, and the 2 independent sets of 23 chromosomes are interchanged. The development of a new human being is underway. Fertilization begins the germinal period. By the end of the first week, the zygote reaches the uterus. The next step is implantation, in which the zygote burrows into the uterine wall and establishes a connection with the woman's blood vessels. Implantation takes about a week to complete and triggers hormonal changes that prevent menstruation, letting the woman know that she has conceived. The implanted zygote is less than a millimeter in diameter, yet its cells have already begun to differentiate. The layer of cells closest to the uterus becomes the placenta, a structure through which nutrients and wastes are exchanged between the mother and the developing organism. After the zygote is completely embedded in the uterine wall, it is called an embryo. This new period typically begins the 3rd week after conception and lasts until the end of the 8th week. During this period, body structures and internal organs develop. A dramatic way to see these changes is to compare a 4-week embryo with a 7-week-old embryo. The 4-week-old embryo on the left is about 2 millimeters long and looks more like a salamander than a human being. But growth and specialization proceed so rapidly that the 7-week-old embryo, shown on the right, looks distinctively human, with eyes, arms, and legs. The brain and the nervous system are developing rapidly, and the heart has been beating for about a month. Most organs found in a mature human being are in place in some form. The final and longest phase of prenatal development is the fetal period, which begins at the 9th week and ends at birth. During this period, the baby-to-be becomes much larger and its bodily systems begin to work. The increase in size is remarkable. At the beginning of this period, the fetus weighs less than an ounce. At 3 months, the fetus weighs about 3 ounces. At about 4 months, the fetus weighs roughly 4 to 8 ounces, which is large enough for the mother to feel its movements. In the last 5 months of pregnancy, the fetus gains an additional 7 or 8 pounds before birth. During this period, genitals form and sex hormones cause differences in fetal brain organization, and this period experiences the greatest brain growth during the 4th, 5th, and 6th months. Interestingly, the fetus can learn and remember. When a fetus is exposed repeatedly to distinct sounds, such as the mother's voice, or distinct tastes, say the mother eats garlic, the fetus recognizes those stimuli after birth. Research has shown, for example, that after women drank carrot juice frequently during the last month of pregnancy, as 5- and 6-month-olds, their infants preferred cereal flavored with carrot juice. These skills are adaptive in helping newborns to recognize mother by her voice and in helping them to learn language because the sounds are familiar. The age of viability is the age at which a preterm newborn may survive outside the mother's uterus if medical care is available. It begins about 22 weeks after conception. By this age, the fetus has a distinctly baby-like look, but babies born this early can have trouble breathing because their lungs are not yet mature. Sometimes inherited disorders, or an unfavorable prenatal life, or a difficult birth affect a person lifelong. Fortunately, healthy newborns are the norm, not the exception. There may be chromosomal anomalies. About once in every 200 births, An infant is born with 45, 47, or even 48 or 49 chromosomes, which produces a syndrome. The most common condition is Down syndrome, trisomy 21. The term trisomy is used to describe the presence of 3 chromosomes, rather than the usual matched pair of chromosomes. If a baby is born with 3 number 21 chromosomes, rather than the usual pair, then the baby would be said to have trisomy 21. People with Down syndrome typically have distinctive characteristics, including unusual facial features, heart abnormalities, and language difficulties. About 1 in every 500 infants is born with only 1 sex chromosome, no Y, or with 3 or more, not just 2. Having an odd number of sex chromosomes impairs cognition and sexual maturation with varied specifics. It's not unusual for affected people to seem to be typical until adulthood, when they consult a doctor because they are infertile. There is a table in the chapter which identifies some common abnormalities involving the sex chromosomes and describes some of the associated physical appearances and psychological characteristics, along with the incidence rates. With gene disorders, the phenotype is only affected when the inherited gene is dominant, when both parents carry the recessive gene, or when additive genes combine to cause a problem. Most dominant disorders begin in adulthood. Fatal dominant childhood conditions cannot be passed on, and recessive disorders are more common. Some examples are Huntington's disease, cystic fibrosis, sickle cell disease, and Fragile X. If parents are concerned about passing on a genetic disorder, they may seek genetic counseling. Professionals who provide genetic counseling help prospective parents understand their genetic risk so that they can make informed decisions, not impulsive, irrational ones. A counselor asks about family medical history and constructs a family tree for each parent to assess the odds that their child will inherit a disorder. If the family tree suggests that a parent might be a carrier of the disorder, blood tests can determine the parent's genotype. With this information, a genetic counselor then advises parents about their choices. A couple might go ahead and conceive a child naturally, use sperm or eggs from a donor, or adopt a child. Prenatal harm can be the result of teratogens, any agent or condition, including viruses and drugs, resulting in birth defects or complications. The following are examples of common teratogens and the defects they can cause. Alcohol is one of the most commonly recognized teratogens. Alcohol damages brain cells, which can lead to intellectual disability in many children who were exposed while in the womb. It can also lead to abnormalities in face and head size, as well as generally slow growth, and cause abnormalities in the rest of the body. Nicotine is another widely recognized teratogen, and pregnant women are strongly encouraged not to smoke. Nicotine exerts its harmful effect on the fetus by constricting the blood vessels, which leads to less blood flow to the baby. Because blood circulating from the mother is the only way the fetus can get oxygen, this leads to hypoxia, or low flow of oxygen, and the fetus effectively suffocates. The result can be low birth weight and even premature birth. These conditions can cause a variety of health problems that may last for the baby's lifetime. Many women take prescription medication during pregnancy for necessary reasons like diabetes, seizures, depression, anxiety, and other medical conditions. Each medication approved by the FDA has a risk factor classification, including potential risk factors during pregnancy. These ratings, categories A, B, C, D, and X, along with an evaluation of the risks and benefits of using a particular medication, will help the mother and the healthcare provider determine what steps to take. Pregnant women should always consult the doctor about their medication. Embryos exposed to a mother's heavy drinking can develop fetal alcohol syndrome, FAS. Fetal alcohol syndrome causes brain damage and growth problems. The problems caused by FAS vary from child to child, but defects caused by fetal alcohol syndrome are not reversible. There is no amount of alcohol that's known to be safe to consume during pregnancy. Alcohol causes higher blood alcohol concentrations in the developing baby compared to the mother's body because a fetus metabolizes alcohol slower than the mother does. Alcohol interferes with the delivery of oxygen and optimal nutrition to the developing baby. Having a healthy pregnancy is one of the best ways to promote a healthy birth. Getting early and regular prenatal care improves the chances of a healthy pregnancy. This care can begin even before pregnancy with a preconception care visit to a healthcare provider. According to the Centers for Disease Control, overall, 77% of women who gave birth in 2016 initiated prenatal care in the first trimester of pregnancy. 4.6% began prenatal care in the third trimester, and about 1.6% of women received no care at all. Advocates for improved maternity care have claimed that the U.S. maternity care system frequently offers fragmented impersonal care. Fewer women are taking prenatal education classes, and more women are experiencing high risk or complicated pregnancies. The total time a woman spends in prenatal visits has been reduced. The typical woman may have as little as 2 hours of interaction with her doctor during her entire pregnancy. These advocates also claim too many women end up with physical and emotional health problems after giving birth. Problems can occur when babies are born too early or too small. Small for gestational age is when birth weight is significantly lower than expected, given the time since conception. Babies born at 35 weeks or sooner are called preterm. Babies with very or extremely low birth weight do not fare well. Special care is needed for these babies, not only in the hospital, but also when they go home. Long-term positive outcomes depend on providing a supportive home environment. A 30-year study on the Hawaiian island of Kauai found that when low birth weight babies grew up in stable homes, defined as two mentally healthy parents throughout childhood, they were indistinguishable from children born without birth complications. The message is that excellent caregiving can compensate for all but the most severe birth complications. Low birth weight is defined as follows: less than 2,500 grams, 5.5 pounds at birth. Very low birth weight is under 1,500 grams, or 3 pounds 5 ounces at birth. Extremely low birth weight is under 1,000 grams, or 2 pounds 3 ounces at birth. The United States has seen a steady increase in low birth weight over the past 25 years. 8% of newborns are seriously underweight and more susceptible to teratogens and higher birth risks. When comparing nations, the United States has a low birth weight rate of about 8%, ranking worse than most developed nations. Worldwide, far fewer low birth weight babies are born today than 20 years ago. Neonatal deaths have been reduced by 1/3 as a result. But what explains the differences in infant birth weights when compared to the United States? Low birth weight can usually be prevented when a pregnant woman gets adequate prenatal care. But many pregnant women in the United States receive inadequate or no prenatal care because they have no health insurance. Virtually all countries that rank ahead of the U.S. provide complete prenatal care at little or no cost. It's interesting that at birth, the baby knows when he or she is ready. The fetal brain signals the release of hormones to trigger the female's uterine muscles. Labor begins. The average duration for firstborn babies, 12 hours, and there's quicker labor for later-born babies. The APGAR scale is a quick assessment of a newborn's body functioning. The baby's heart rate, respiration effort, muscle tone, color, and reflexes are given a score of 0, 1, or 2 in each category twice, at 1 minute and 5 minutes after birth. And each time the total of all 5 scores is compared with the ideal score of 10, which is rarely attained. Most babies score at 8 or higher. An APGAR score below 7 is indicative of an emergency, needing immediate attention from a neonatal pediatrician. When a normal, uncomplicated birth is not possible, there's a need for medical intervention. A C-section may be performed. This is a surgical birth, and the fetus can be removed quickly. It's usually safe for mothers and babies, saving lives when the fetal head is too large for the pelvis. When a fetus is in distress, or when the fetus is in an irregular position, a physician may decide to remove it through a C-section. A C-section is riskier for the mother than a vaginal delivery because of increased bleeding and a greater danger of infection. A C-section poses little risk for babies, although they are often lethargic briefly from the anesthesia that the mother receives before the operation. Now, there are lower rates of C-sections in poorer countries and higher rates in richer countries, usually planned for the convenience of the mother and or the obstetrician. And 1/3 of births in the United States are through C-sections. When I was born in the late 1950s, women in labor were admitted to a hospital and administered a general anesthetic. Fathers waited anxiously in a nearby room for news of the baby. But childbirth has changed. Today, many varieties of prepared childbirth are available to pregnant women. These share some fundamental beliefs. One is that birth is more likely to be problem-free and rewarding when mothers and fathers know what is happening. So they go to classes to learn basic facts about pregnancy and childbirth. Another belief is that natural methods of dealing with pain are better. Consequently, pregnant women learn deep breathing methods which help them relax to reduce pain. A third thing is to have a supportive adult present to provide emotional support and help a woman use techniques for managing pain. There is a trend in natural childbirth to have babies at home. For Americans accustomed to hospital delivery, home delivery seems risky, and some medical professionals remain skeptical. A trained birth assistant, known as a doula, may be present. Humans are social creatures and seek interaction, so consequently a professional might administer the Brazelton Neonatal Behavioral Assessment Scale to help parents interact with their newborn. It looks at key developmental tasks: control of autonomic and motor systems, state of consciousness, and social interaction. It gives parents guidance on how to support their newborns. For example, a newborn struggling with motor control might need more holding and soothing in order to settle down. Or a baby that is struggling with social interaction might need to be presented with one face to focus on at a time, and not a room full of faces at once, or even one immediately after another. For parents, the time immediately after a trouble-free birth is full of excitement, pride, and joy. The baby is finally here. But it is also a time of adjustment. Parents must adjust psychologically as they reorganize old routines to fit the baby's sleep-wake cycle, for example. One complication for mothers is postpartum depression. Many women experience significant physical problems after birth. Psychological symptoms range from baby blues to postpartum psychosis, and they may involve struggles with providing adequate baby care. There are various causes of postpartum depression. Changes in hormonal levels following birth place some women at risk. Women are likely to experience postpartum depression when they are single, were depressed before the pregnancy, are coping with other life stresses, such as dealing with the death of a loved one, did not plan to become pregnant, or did not have other adults to support their adjustment to motherhood. Pregnancy can seem like it's all about the mom, but it is so important for dads to be part of the experience. Most fathers are helpful to their pregnant wives. They help the mother stay healthy, and they help the mother to manage stress, which can affect the fetus. Pregnancy emotions will definitely run the gamut: ups and downs, highs and lows, excitement and dread. The bottom line, fathers need to be there through it all. Sympathetic pregnancy, KUVAD, describes a situation in which otherwise healthy men whose partners are experiencing pregnancy experience pregnancy-related symptoms themselves. While some research suggests that KUVAD might be common, it isn't a recognized mental illness or disease. A range of theories have been proposed to explain the Kuvad syndrome. Along with psychoanalytical and psychosocial explanations, there are explanations that include emotional attachment to both the unborn child and partner. Kuvad is expected in some cultures, a normal variation in many cultures, and considered pathological in others.